India, and I'll be moderating this talk with Swagatama Mukherjee, who's from MS University of Baroda. On behalf of SNEV, I extend a warm welcome to today's speaker, Dr. Manuel Varas-Godoy, and to everybody else in the audience who have tuned in for this exciting talk today. Now, I'll briefly introduce today's speaker to the audience. Dr. Manuel Varas-Godoy is a biochemist from the Pontifical Catholic University of Valparaiso in Chile and a PhD in biotechnology at Andres Bello University in Chile. During his PhD, he studied the effects of a family of non-coding mitochondrial RNAs on cancer development using a melanoma model. Later, he completed his postdoctoral degree at the Karolinska Institute in Sweden, and here his work was focused on how calcium signals could regulate the progression of bladder cancer. On his return to Chile in 2015, he joined as a young researcher in the reproduction biology laboratory of the University of Los Andes to begin his independent research in the field of tumor extracellular vesicles. Currently, he is an associate professor at the Faculty of Medicine and Science and principal investigator at the Center of Cell Biology and Biomedicine at San Sebastián University and an associate investigator at the BASAL Centro Ciencia Vida. Today, Dr. Varas-Godoy will be talking about the biogenesis and secretion of pro-tumorigenic extracellular vesicles. A note to the audience members, please put your questions in the chat box. We'll be going through them at the end of the talk. The stage is yours, Dr. Varas-Godoy.
Thank you, Ayuj, and thanks to all the people present here in this seminar. First of all, I appreciate this invitation because I think this is a really good network. How do you say? First of all, sorry for my English because I'm a little bit rusty, but I will try to do my best. But continues my presentation. I want to say thank you to all the organizers, Cristobal, Ayuj, Swatama, for inviting me. And I want to present what we are doing here in our lab. And the title of the presentation is Different Stimuli Have the Same End, Biogenesis and Secretion of Pro-tumorigenic Extracellular Vesicles. I want to show where is my place that I work. Here you can see our university, Universidad San Sebastián. He's in the middle of the downtown here in Santiago de Chile. Yeah, Santiago de Chile. You can see it's a really nice place that is like a microenvironment in a big city. Yeah.
Sorry to bother you. Your presentation is not sharing right now on the screen.
Oh, sorry.
That's okay.
Sorry. Now?
Yes, it's perfect.
It's good.
Yeah. Perfect. Sorry. So, as I told you, this is the title of my presentation. But first of all, I want to present where I work. Yeah, here's the Universidad San Sebastián. It's in the middle of the city. You can see here. Yeah. And it's like a microenvironment in the middle of the city. It's a really nice place. This is our building, the Center for Cell Biology and Biomedicine. But in a couple of months, we will move to another place here. So all of you are invited to visit me if you want to come to Chile. And as this microenvironment, we work in our lab in the tumor microenvironment. I love this picture because I think demonstrate all the process that can participate in tumor progression. You can see here the tumor, and of course, in the tumor and the tumor microenvironment, there are different type of cells. You can see here, for example, mesenchymal stem cells, or cancer-associated fibroblasts, dendritic cell, among others. But also, you can have in the tumor microenvironment proteins like a soluble factor, TGF-beta, PTGF, Wnt, for example. You can have the matrix, the extracellular matrix, that also is considered part of the tumor microenvironment. And in the last years, not last year, but 10 or 20 years, we know that there are another participant in all this tumor microenvironment called extracellular vesicles. Here we can see one of type of the extracellular vesicle, exosome, that can participate in the tumor microenvironment to promote tumor progression, including all the process related to metastasis, intravasation, survival in circulation, extravasation, and preparation of the pre-metastatic niche. So in our lab, we work in that field, in how component of the tumor microenvironment can regulate the biogenesis and secretion of extracellular vesicles. And you know better than me that extracellular vesicles can participate in different process related to tumor progression, like angiogenesis, the preparation of pro-metastatic niche, genomic instability, resistance to cell death, among others. So all these processes are related to tumor progression. And in that context, in the tumor microenvironment, the tumor can be exposed to different stimuli. For example, we can see thermal stress, hypoxia, radiation, difference in the pH, or even drug stimulation. You know that the chemotherapy also will affect the tumor. So all these stimulus can trigger biogenesis and release of extracellular vesicles. So in our lab, we work, for example, with drug stimulation. And it's known that chemotherapy can promote release of extracellular vesicles. Here we can see this paper that the massive release of extracellular vesicles from cancer cells after photodynamic treatment or chemotherapy. But there are several papers showing that. Now, you can see here, and you can see here. So this stimuli, like chemotherapy, can regulate the release of extracellular vesicles, but also can participate in the as a mechanism of chemoresistance that is mediated by extracellular vesicles. Here we have three mechanisms that extracellular vesicle participates in chemoresistance. Here, for example, extracellular vesicle released by the donor cells, in that case the tumor cells, can eliminate the drug into the extracellular vesicles, or also they can transfer proteins to a recipient cells, for example, the chemoresistant cells to sensitive cells. In this case, transfer efflux pumps that can transfer the chemoresistance, or can transfer another type of macromolecule, microRNAs, or protein that promote cell proliferation, decrease apoptosis, inactivate the drug, or protein that participate in DNA damage repair. So in our lab, we did a question that how chemoresistance is involved in biogenesis and release of extracellular vesicles. And if you go to evaluate which process are involved in the secretion and biogenesis of extracellular vesicles, especially exosomes, you can see here, yes, the early endosome that maturate to late endosomes or multivesicular bodies. And these have two roads to take. Go to degradation or fusion with the plasma membrane to release the exosome. And in all these steps, you have different proteins, as you can see here. You have one step, which is the formation of the interluminal vesicles. You have another step when you have the transport of the multivesicular body to the plasma membrane or to the lysosome or autophagosome, and proteins related to the fusion of the multivesicular bodies with the plasma membrane or with the autophagosome on the lysosome. So in that context, we use this chemoresistant cell model that is an ovarian cancer cell called A2780. And when you're exposed to a chemotherapy like cisplatin for several months with increasing amount of the drug, you can create these chemo-resistant cells. Just to compare, you can see here the LD50 of the cells, and when you have the sensitive cells, the LD50 is 0.85 or 1 micromolar around, and the A2780Cs that are the cell resistant to cisplatin have an LD50 of 12. So increase a lot the chemo-resistance against the chemotherapy. And what we did was we can see all these components that we saw first. We can evaluate what happened with this component in these cells. And first of all, we evaluate multivesicular bodies. And how we evaluate that? With a marker of multivesicular body that is CD63. And we did immunofluorescence in these cells. We can see more structure related to CD63 in the resistant cells in comparison with the sensitive cells. And here is the quantification. We have more structure per cell, and also the area of the CD63 structure are bigger in comparison with the sensitive cells. Super. So after that, we did a electron microscopy of this structure. We analyzed this structure in comparison the resistant against the sensitive cells, and we can see more multivesicular body or multivesicular endosome in the resistant cells in comparison with the sensitive cells. The area also is bigger, and the number of interluminal vesicles inside of the multivesicular body is higher. So seems that our chemo-resistant cells have more multivesicular bodies. So after that, we evaluate several proteins related to the biogenesis of exosomes, like HRS, that is a component of the SCORE machinery, ALIX, TSG1, DS101, that all of them participate in the SCORE machinery, and LAMP2A that the last year was involved in biogenesis of extracellular vesicles. And you can see that in the resistant cells, all the proteins have more levels in the resistant cells in comparison with the sensitive cells. So seeing that we have more machinery to promote the biogenesis of exosomes. But one of the steps is biogenesis. The other step is transport of the multivesicular body. And we evaluate different proteins related to the transport to the multivesicular body to the plasma membrane. Here we evaluate RAP11, RAP22, RAP27A, and RAP35. And you can see that only RAP27A increase the level of proteins in the resistant cells in comparison with the sensitive cells. And here you can see the quantification, and this increase is huge. We can see almost 6 times of increase in the protein level of RAP27 in the chemo-resistant cells. So we have more proteins to biogenesis, and this impact in have more multivesicular bodies, I guess. And also we have this increase in the RAP27A, so we have more transport to the plasma membrane of the multivesicular body. So we check now what happened with the secretion of extracellular vesicles. And if we compare the resistance cells with the sensitive cell when we isolate extracellular vesicles, here is the characterization. We have the markers of the extracellular vesicles or exosome as a CD63, TSG101, or synthenin. And we can see these markers in the extracellular vesicles enriched in comparison with the cell lysis. We can see also the electron microscopy, and the extracellular vesicles have the same morphology. But when you compare here the secretion, you don't have more secretion of extracellular vesicles from the resistant cells in comparison with the sensitive cells. You have more secretion only when you treat the cells with the chemotherapy. You can see here that we have more secretion just in the cells that have more the cells that were treated with cisplatin in comparison with the resistant cell without treatment or the sensitive cells. So just when we treat the cells with the chemotherapy, we have more secretion. And as I told you, these extracellular vesicles is known that can transfer chemoresistant to a sensitive cell. So we did an experiment that where we culture sensitive cells with the different extracellular vesicles. Without extracellular vesicles isolated from sensitive cells, extracellular vesicles isolated by the chemoresistant cells, and extracellular vesicles isolated from the chemoresistant cell, but in response to cisplatin. And we wait 16 hours, and after that, these cells treated with extracellular vesicles were exposed to cisplatin, and 48 hours later, we did the cell death analysis. And you can see here, using two techniques, flow cytometry, light death staining, and the typical trypan blue staining. And in both cases, the only cell, sensitive cells that acquired chemoresistance were these cells that were treated with extracellular vesicles derived from resistant cells in response to cisplatin, in comparison with the resistance, with sensitive extracellular vesicle, or without treatment. And in both cases, we had the same result. So yes, these cells secrete extracellular vesicles that can transfer chemoresistance, but only in response to cisplatin. So the cells have more machinery, seems, but need the stimuli to secrete this pro-tumorigenic extracellular vesicle. We did an spectromic analysis of the proteomic analysis of these extracellular vesicles. We found that are super enriched in proteins related to repair the DNA double strand, to stabilize p53, regulation of RNA2, regulation of p10, and a lot of protein related to metabolism of RNA. So, and all these protein in this interactomic participate in the transfer of chemoresistance. But the extracellular vesicles, we saw that we have in the chemoresistant cells more machinery to have multivesicular bodies and to transport the multivesicular bodies to the plasma membrane, because we have a high level of RAC27. But what happened with the other road, the degradative road, the road to the lysosome? So what happened with lysosome in these cells? And you can see here that the resistant cells have less protein level of LAM1, this typical protein of lysosome, and we can see by Western blot, and here is the quantification. And also we did immunofluorescence against LAM1 and cathepsin D, and in both cases, in both proteins are less in the resistant cells in comparison with the sensitive cells. This thesis is this work was performed by Cristóbal Cerda, and this was published the last week in Journal of Cellular Biology. And here we have the quantification of the immunofluorescence. So we have less lysosomes, but what happened with the activity of the lysosome? And we did this staining with magic red that measured cathepsin B, and the resistant cells have less lysosomal activity in comparison with the sensitive cells, but also decrease RAP7. So you can see that the resistant cells have more RAC27, that is the protein involved in the transport of the multivesicular body to the plasma membrane, but have less level of RAC27, that is protein involved in the fusion of the multivesicular body with the lysosome. So seems that the resistant cells have more preference to secrete in comparison to degrade. So what happened now with the lysosomal activity if we block the exosome secretion? Because as I told you, we have all this balance to the exosomal secretion. So what happened if we block the exosomal secretion? So we did a knockdown of RAP27, this protein that we saw increased 6 times in the resistant cells. You can see here that here we have the increase in the resistant cells, and when we do the knockdown with an shRNA of RAP27, we decrease the level, and here is the quantification, and see what happened with RAP7. Recover its level. So when we block RAP27, we increase the level of RAP7, but also we increase the level of LAMP1, this protein of the lysosome. So the knockdown of RAP27A recover protein level of RAP7 and LAMP1 in the resistant cell. So it seems that now we block the secretory system of extracellular vesicles, and the cell recover all the degradative system. We measure again LAMP1 and cathepsin D by immunofluorescence, and you can see here that when you block the secretion of exosomes, the LAMP1 and cathepsin D levels are recovered. We saw also the lysosomal activity, and also is recovered by magic red. So the question now is, what happened with the exosome secretion if we increase lysosomal activity the other way? First of all, we block the exosome secretion, but what happened now if we increase in the resistant cell the lysosomal activity? For that purpose, we use rapamycin, that is known that inhibit the mTORC1, and with that we can promote the lysosomal biogenesis. So you can see here in the different group the phosphorylation of the protein S6K that is activated in response to cisplatin, but when we use rapamycin, this is blocked. And you can see here the activation of mTORC1 and the inhibition with rapamycin. And after that, we isolate the extracellular vesicles, yeah, and we did Western analysis, yeah, and NTA. Here, just to check, with rapamycin, we increase the level of LAMP1 and cathepsin D in comparison with the other cells. So we are increasing the lysosomes. And when we did the analysis of extracellular vesicles, you can see the effect of cisplatin in the resistant cell that we saw first, that increased the secretion of extracellular vesicles, but rapamycin, so the block of mTORC1, decrease the level of exosome secretion in response to cisplatin. So, a summary, we have an increase of the degradative, of the secretive system of extracellular vesicles, decrease of the degradative system in these resistant cells, and when we block the secretion, we increase the lysosomal activity, or when we increase the lysosomal activity with rapamycin, we decrease the exosomal secretion. So seems that there are a balance between lysosomal activity and exosome secretion. But now we check what happened with the activity of the exosomes related to the transfer of chemoresistance. So we isolate the extracellular vesicles from all the group, and we culture the extracellular vesicle with the Ad2780 sensitive cells, the same experiment that we showed before. We treat it later with cisplatin, and we did the analysis of cell death. And you can see here that in orange, we have this transfer of chemoresistance promoted by the cells, the extracellular vesicles, in response to cisplatin. But when we block with rapamycin, mTORC1, so we increase the lysosomal activity and decrease the exosomal secretion, this extracellular vesicle also lost the capacity to transfer chemoresistance in both cases. So it's not just they live to secrete extracellular vesicles. They also change the activity of these extracellular vesicles. So as a summary of the first part of the talk, we saw that these extracellular cells that are sensitive to cisplatin have more lysosomes, more lysosomal activity. And when they transform to chemioresistant cells, this change this lysosomal activity, and now they have less lysosomal activity, but have more capacity to generate extracellular vesicles and secrete extracellular vesicles in response to cisplatin. And when we block the secretion of extracellular vesicles, or we increase with rapamycin the biogenesis of lysosomes, so we recover the lysosome activity, they acquire again a sensitive phenotype. So we have this balance between secretory and lysosomal activity. So this was the end of this story, but why is there only implicit secretion in response to cisplatin? Because the resistant cell only secrete extracellular vesicles in response to cisplatin. So we did more analysis of another component of the exosome secretion, and we found several proteins related to the SNER complex increased in the cells, resistant cells in response to chemotherapy in comparison with the resistant cells without treatment. So now we are doing the analysis of this protein that is part of this SNER complex. So this is the thesis of Alvaro Neira of our lab. So we hope that have a follow-up story to show you in the future. But in the lab, we also work with another stimuli, not just drug stimulation. And one of the stimuli that is present in the tumor microenvironment is hypoxia. So we know that hypoxia is involved in EV biogenesis and release. And you can see this picture that hypoxia can induce MBB transport to the plasma membrane. So they can induce fusion of the multivesicular body with the plasma membrane. And we have an increase of RAP27, you can see here, and decrease of RAP7. The same effect that we saw with the chemoresistance. And also they can induce lysosomal dysfunction. The hypoxia also induces lysosomal dysfunction. So for us also it's important to study this type of stimuli. But not only change the endosomal traffic, they also change the cargo of the extracellular vesicle. And there are several publications that show that extracellular vesicle derived from hypoxic cells change its cargo. For example, you can see here, like these extracellular vesicles have several cytokines, like interleukin-6, secretion, have even HIF1 alpha, some microRNAs, among others. And all these cargo are involved in promote pro-tumorigenic properties like angiogenesis. So we wanted to study what happened in hypoxia, and this is why our cellular model, we use mainly this adenocarcinoma, lung adenocarcinoma cell line, call it A549, and we use this condition of hypoxia, 1% of oxygen, and we compare against normoxic cells, culturing in a standard condition, and we isolate by ultracentrifugation the extracellular vesicles, what we call tumor small extracellular vesicle hypoxic or normal. We characterize the extracellular vesicles, and they have the typical markers, ALEX C63, the typical morphology, and they have around 150 nanometers of diameter. So perfect. And we transfer these cells, as I told you, we know that extracellular vesicle derived from hypoxic tumor cells can induce angiogenesis. So we measure two properties related to angiogenesis, like migration of endothelial cells and tube formation of endothelial cells. We use this cell line, call it HI 926, and we culture the cells with 5,000 small extracellular vesicles derived from hypoxic or normoxic condition. And you can see here that the extracellular vesicles derived from hypoxic condition promote more migration measured by Transwell in comparison with the normoxic extracellular vesicles, or in comparison with the non-treated cells. And also we saw more formation of tubes in vitro in the cells cultured with the hypoxic extracellular vesicles in comparison with the normoxic. This is known in the literature. So we try to know what is the mechanism involved in the induction of pro-angiogenic properties induced by tumor small extracellular vesicle derived from hypoxic condition. And one of the systems that, or the cells, the pathways that is known, that is involved in the induction of pro-angiogenic properties is the beta-catenin pathways. Donde you can see here that beta-catenin, when it's not activated, is sequestered by this complex of obstruction and is degraded to the proteasomal, is phosphorylated and degraded for the proteasomal degradation. But when this is not phosphorylated, this can go to the nucleus and activate this transcription factor called TCF-LEF and promote the expression of different genes involved in pro-angiogenic properties like MMP2, VEGF-A, among others. And it's known that extracellular vesicles can activate beta-catenin pathways. So we measured that, and we saw that the extracellular vesicles cultured with the recipient cells, T-high cells, the endothelial cells, promote the nuclear localizations of beta-catenin in these endothelial cells. And you can see here, this picture is the endothelial cells cultured with the hypoxic extracellular vesicles, and we saw more localization of beta-catenin in the nucleus in comparison with the other condition. We did a nuclear fractionation of cells, and we have the nuclear fraction and the cytosolic fraction, and we can see that beta-catenin is more present in the nucleus, in the total or non-phosphorylated beta-catenin is more located in the nucleus in comparison with the other condition. We saw if beta-catenin is active in these endothelial cells cultured with hypoxic extracellular vesicles. And here we use a reporter analysis using this top renila reporter. And the endothelial cells have, this top renila reporter is the reporter of the TCS-LEF transcription factor. And you can see here that the extracellular vesicle derived from hypoxic condition promote the activation of TCF-LEF in comparison with the other conditions. And we saw target of TCF-LEF like MMP2 on VEGF-A, and we saw that the extracellular vesicle derived from hypoxic condition promote the expression of MMP2 and VEGF-A in comparison with the other conditions. Perfect. We saw if the effect of extracellular vesicle in the pro-angiogenic properties is because the beta-catenin is activated. So for that reason, we use an inhibitor of the translocation of beta-catenin to the nucleus, that is C59. And also we use a small interference RNA against beta-catenin. And we saw here the effect of the hypoxia extracellular vesicle that activate the migration of the endothelial cells. But when we block it with this two approach, beta-catenin signaling, the effect in the migration is blocked. So the tumor extracellular vesicles promote endocellular cell migration via beta-catenin signal. One, to activate the beta-catenin signaling, need the stabilization and the nuclear localization of beta-catenin. And for that, we need an endosomal sequestration of the destruction complex of beta-catenin. And this endosomal sequestration is in the early endosome. So we need to have more early endosome and activation of RAP5 in the cells to have sequestration of beta-catenin to the destruction complex. So we did the analysis of what happened with the early endosomes and beta-catenin in the destruction complex. And you can see here that the hypoxic extracellular vesicles promote the sequestration of component of the destruction complex like GCK3 beta to the endosome, the early endosome, measured here with the protein EA1 in comparison with the other condition. So seems that the destruction complex of beta-catenin is recruited to the early endosome. We measure this component and also this component of the destruction complex called axin, and in both cases, this correlate with the early endosome. So they colocalize with early endosome. So seems that the destruction complex is recruited to the early endosome in endothelial cells when they are treated with extracellular vesicle derived from a hypoxic condition. So what happened with the early endosome, because they need grow if they are activated. So we measure EA1, that is a marker of the early endosome, and we can see that we have more structure EA1, positive EA1, and also we have an increase in the size of this endosomes compartment when we treated the endothelial cells with hypoxic extracellular vesicles. We did the same analysis, but measuring RAP5, that also is located in the early endosome, and we saw the same. Hypoxic extracellular vesicles promote the increase in this type of structure and the size of this type of structure. If we have an activation of the early endosome to recruit the destruction complex of beta-catenin, we need activation of RAP5. So we measure the activation of RAP5. So we did the pull down of an active RAP5 that is bind to GTP, and we can see that in the cells we have more RAP5 active when we treat it with hypoxic extracellular vesicles in comparison with the normoxic. And we saw if the effect in the pro-tumorigenic properties is RAP5 dependent. So what we did was transfer the endothelial cells with the negative, with the dominant negative RAP5, and we saw that the cell migration of endothelial cell increased with the hypoxic extracellular vesicle, but when we block RAP5, this increase is blocked too. So the tumor, the hypoxic extracellular vesicles promote endothelial cells, and this is dependent of the RAP5 activation. Some years ago, with Vicente Torres, we published this article that hypoxia promote the expression of alsin-2. And alsin-2 is a RAP5, is a gift of RAP5. So promote the activation of RAP5. So and in the article what we published some weeks ago, we check that the alcin-2 in these cells in our model increase in response to hypoxia, and in fact they increase in hypoxia in comparison with the normoxic condition. But more interesting, we saw that the extracellular vesicle derived from the hypoxic condition have is loaded in the extracellular vesicle, and we have more levels of alcin-2 in this extracellular vesicle in comparison with the normoxic extracellular vesicle. And here we have the characterization of the typical protein in the extracellular vesicle, synthenin, aligan, CD63, and the negative control. So, and when we culture the endothelial cells with these extracellular vesicles, we saw that they alcin-2 colocalize with the early endosome. We can see here the EA1 and alcin-2, and we saw the increase in the colocalization of alcin-2 with EA1 structure in comparison with the other condition. So it seems that the alcin-2 loaded in the extracellular vesicles promote the activation of early endosomes. So we asked the alcin-2 if alcin-2 loaded in the hypoxic extracellular vesicle contribute to promote these pro-tumorigenic properties. And what we did was use the negative dominant of alcin-2. Here we have the effect of immigration of the endothelial cells, and as we saw previously, hypoxic extracellular vesicles promote the migration of endothelial cells in comparison with the normoxic extracellular vesicle, but this increase is blocked when we use we transfect the endothelial cells with the dominant negative of alcin-2. So it seems that alcin-2 loaded in the extracellular vesicle could be involved in the promotion of the pro-tumorigenic properties. To continue this work, we did a knockdown, a silences of alcin-2 using an shRNA against alcin-2. Here we can see the cells that in hypoxia increase the level of alcin-2, and in normoxia decrease the level of alcin-2. The shRNA, this is the control of our shRNA, and also change the pattern of the alcin-2 in the extracellular vesicle. Here we have more alcin-2 in the hypoxic extracellular vesicle in comparison with the normoxic, but this increase is blocked when we isolate extracellular vesicles from the cells with the shRNA against alcin-2. And we measure if these extracellular vesicles promote the effect that we saw before. So we did the analysis of the activation of RAP5, and you can see here that the activation of RAP5 is not induced by the extracellular vesicles derived from a hypoxic or normoxic condition of the extracellular vesicles isolated from the tumor cells with the silencer of alcin-2. Also, the increase in the early endosomes also was blocked when we use the extracellular vesicle derived from the cells from the shRNA of alcin-2. Remember that beta-catenin was translocated to the nucleus when we culture the cell with the hypoxic extracellular vesicles. So we measure the same here, and when you can see here the quantification, we have the increase in the beta-catenin localization, nuclear localization in the cells cultured with the hypoxic extracellular vesicle, but this effect is lost when we culture with the extracellular vesicle derived from hypoxic condition, but with the silencing of alcin-2. We measure now the activity, if beta-catenin is active when we culture with the extracellular vesicle. Here in white, you can see the effect that we saw previously, that we have more TCF-level activity, and we have more expression of MMP2. Here is the VGFA, it's not MMP2, sorry. And this effect is blocked when we silence the shRNA from the secretory cells. So seeing that alsin-2 in the extracellular vesicles is needed to activate beta-catenin pathway. And finally, we measure what happened with the pro-tumorigenic activity and the migratory effect and the tube formation assay that increase in the endothelial cells culture with the hypoxic extracellular vesicle is lost when we use extracellular vesicle derived from the hypoxic cells with the silencing of alsin-2. So we propose the following models that we have these donor tumor cells. We prove in another cell like MCF7 and MDA231 that are breast cancer cells, and we saw the same effect. And when we put the cells in hypoxic condition, increase the level of alsin-2, this alsin-2 is loaded in extracellular vesicles, transfers to endothelial cells, and these endothelial cells activate the early endosomes. And this activation of the early endosome preclude the destruction complex of beta-catenin. And beta-catenin can translocate to the nucleus and promote the expression of genes that are related to tube formation and cell endothelial migration. The story continues, and of course, we want to know what happened with the donor cells in its endosomal trafficking, because we know that alsin-2 increase in the tumor cells. So we want to study what happened in this endosomal traffic and if the expression of alsin-2 is involved in that the multivesicular bodies can prefer fusion with the plasma membrane and not go to the lysosome degradation. So this is the thesis of Belén Gaete that we are working now. And of course, this work is done by other person, not by me. So all the time, I want to acknowledge all the people of the lab that you can see here, especially Belén Gaete, that work in both publications, but also our collaborators. That is the lab of Patricia Burgos and Eloisa Arias that work hard in this, the first manuscript. Matías Ostrowski, that is a collaborator from Universidad de Buenos Aires, and a lab of Vicente Torres and Patricio Silva, that they are one of the first author of the manuscript related to alsin-2. Rodrigo Acuña, another collaborator from Universidad del Desarrollo here in Chile, and the ex-member of the lab, especially Cristóbal Cerda, that is the first author of the first history related to chemo-resistant extracellular vesicles. And that's it. Thank you for your attention.
Thank you, Dr. Varas. This was a wonderful talk. And I'm pretty sure the audience members will definitely have some questions to ask. But let me get the ball rolling with that, actually. So I just have some very basic questions from what I understood. So are you looking for any specific subpopulation in EVs? Are you screening for only MVVs or specifically or small EVs? So, or is it like you just going for a general extraction of all EVs and then specifying only one particular subtype? So what would that entail in this project?
Yes. According to your question, we didn't separate subpopulation. So for that reason, we call it small extracellular vesicles in the publication. Yeah, because we can't separate the extracellular vesicle derived from the exosome, also the endosomal pathway, or the the extracellular vesicle ectosome derived from the plasma membrane.
Right.
But we modulate in both cases pathways related to the exosome secretion. Multivesicular bodies, in the first case with this balance with the lysosome, or in the second case, we increase the activity of the endosomal root with the alsin-2. So we believe that we are affecting the exosomes more than ectosomes derived from the plasma membrane, but we don't know if we also are affecting the secretion of ectosome from the plasma membrane.
Okay, all right. Okay. And my other question is, mostly, I think you focused on the SCART dependent pathways of EV release. Can you comment on, if we look at the SCART independent pathway, how this entire story would play into that?
Yeah, we measure some proteins related to a SCOR dependent pathway. Right. But we saw in the chemoresistant cell, for example, that we have more synthenin. So we believe that maybe in that case, we also have this. It's not a SCOR independent, but it's intermediate SCOR pathway, like the synthenin syndicam pathway. Yeah, and also we are trying to see another independent pathway of the SCOR. Yeah, but seems that almost all the proteins, HIRS, ALIX, and TCAG101 increase in response to chemotherapy. So more than is one pathway, another pathway, seems that chemotherapy increased several pathways involved in secretion of exosomes.
Right. Okay. Thank you. And we have another question here. So Femina is asking, I would like to have your opinion on this. What I have seen from my EV proteomics experiment is that EV carboproteins are majorly enriched in proteins involved in its biogenesis. Are they involved in inducing different functions attributed to EVs for creating pre-metastatic niche or EV coronas, the proteins on EV surfaces that take up to its release from the cells? Is that also involved in this? And her second question is that, did hypoxia increase the EV concentration in your experiment? Because I have worked on hypoxia-induced EVs, but EV concentration by NTA did not significantly alter in my experiment.
Yeah. Can you repeat the first question, please? The second.
Yeah. So the first question is, she's asking that EV, I'm assuming, sorry. So I have seen that from my EV proteomics experiment that EV carboproteins are majorly enriched in proteins involved in biogenesis. Are they also involved in inducing different functions attributed to EVs for creating pre-metastatic niche or EV corona? Is that more involved in this?
Yeah, maybe I don't know if let me see if I understand the question. The proteomic, his or her proteomics is full of protein related to EV biogenesis.
Yeah. That's
And he or she asks if this protein can be involved in the effect in the recipient cell, for example, in the preparation of the
pre-metastatic niche. Yeah,
could be because we saw that in that case that in the second history, that is alsin-2, because that is a protein involved that is a GEF of RAP5, so activate the endosomal traffic, change the activity of the endothelial cells. Yeah, so maybe if you increase, if you transfer protein related to the EV biogenesis to the recipient cells, maybe these recipient cells also activate, in this case, the endosomal pathway. Maybe activate the increase of extracellular vesicles from the recipient cells, and this can prepare the pre-metastatic niche, but now releasing proteins from, or releasing extracellular vesicles from the cells in the pre-metastatic niche. So you can have maybe an active secretion of exosome just for transfer of protein related to exosome biogenesis.
Right. And the second question is, did hypoxia increase the EV concentration in your experiment?
Yeah. In our adenocarcinoma lung model, they didn't increase. But when we saw the other, the MDA cells, the breast cancer cells, they increase the secretion of extracellular vesicles. So I think that depend of the cells and depend of the how aggressive is the cells, because we compare the MCF7 and the MDA secrete more in hypoxia, but the MCF7 doesn't decrease a lot. So I think depend of the cellular model.
Yeah, that is true.
We didn't see increase in the secretion, but we saw that we have the difference, at least in this cargo protein that is alpha-2.
Right. Yeah, I think it also does depend on the disease model, like you mentioned, because I work with glioblastoma and various literature that does suggest that hypoxia, which is a very important factor in GBM, that does play a role in increasing the EV number. So it does depend on the disease model to an extent. And another question that I had is, when you were working with the endothelial cell model, did you look into any of the proteins which are sort of present on the EV surface and aid in VEGF uptake? For instance, if you check string pathway analysis of VEGF, one of the close clusters that come up, they have NRP1, PCAM1, FLP1, which are also have been reported to be seen on the EV surface. So do you have some idea with that?
And in the history of endothelial cells with Alchin-2, we don't have the proteomic analysis, but in the first history with the chemo-resistant cell, we saw in the proteomic analysis that we have a change in the integrin patterns.
Right.
So in the sensitive cells, we have some integrin patterns, some integrins, and in the resistant, we have another pattern of integrin. And we believe that this is because the extracellular vesicles derived from the sensitive cells have one function. In fact, we have a preliminary data that the extracellular vesicles derived from sensitive cells are uptake with a better yield in endothelial cells in comparison with the resistant cells. So they have different targets. Okay. The sensitive cell, we believe that have one target that could be endothelial cells, and the resistant cells have another target that these cells are from ovarian cancer, that these cells could be maybe the cell from mesothelium.
Got it. And have you, could you comment on whether you did any work correlating the chemo-resistant cell population and checking for anoic resistance in those cells? Because usually, could you also comment on if chemo-resistance had any impact on anoic resistance in your particular disease model? Like,
if my extracellular vesicles have another effect in another pro-tumorigenic property?
No. In anoic resistance. So that is, the cell is not dying when it's detaching from the membrane or from the matrix. Yeah. So does that have any correlation with chemo resistance? Because lipid drafts and usually integrins play a major role of that. And I was wondering that if you're looking at chemo resistance and EVs, we definitely could be looking at other factors of the overall cell morphology as well. So can you comment on that as well?
Yeah, our cells, the chemo-resistant cells are not dying. On in hypoxia, we did the experiment, and also they are not dying.
Okay.
Yeah, they are not dying. If you saw in the chemo-resistant work, we didn't use sensitive cells in response to the chemotherapy, because 50% the concentration that we use to promote the secretion of extracellular vesicles is the LD50 of the sensitive cell. So for sure, 50% of the cells will die if we use this. But there are some experiments that people use high concentration of extracellular of chemotherapy, and for a short time, so the cell didn't die, and they don't transfer the chemo-resistant, but the chemo-resistant cells, the extracellular vesicle derived from chemo-resistant cells, transfer the chemo-resistant. So seems that is the chemo-resistant cells have the capacity to secrete extracellular vesicles to transfer chemoresistance and not the sensitive cells.
Right. Got it. Yeah, that's actually quite a breakthrough in the field. And we have another question here, that is, to what extent the degree or severity of hypoxia induction can affect the EV biogenesis and cargo?
Yeah, I think hypoxia, yeah, we have a 1% touch of hypoxia, that is 1%, yeah? And we know that in the tumor, we have different level of hypoxia, depending if we are more in the external part or in the middle of the tumor. So I believe, because I don't know, that depending on the severity of hypoxia, we will have different biogenesis and different cargo and different release. What I can say, but not with hypoxia, but with the chemotherapy, that if we use more, we have this data, but if we use a higher concentration of chemotherapy, the secretion is higher.
Okay.
So I believe that if we have less or higher hypoxic condition, maybe also we will have more secretion or biogenesis or change of cargo.
Sure, right. That's, yeah, one can actually think in that direction. And are there any more questions, Dr. Varas? I would like to request the audience before we move to closing this wonderful talk, actually.
Thank you, thank you. Sorry for my English. I was a little bit rusty.
Oh, no. This was perfectly fine. Actually, I mean, this was a great talk for me because I'm kind of working in the similar field. So it was actually enlightening that the discoveries that I'm working on, it's not too far off and I'm not in the wrong direction. So great talk,
Perfect.
Dr. Varas. And if there aren't any more questions, we can move on to our special announcement. We actually have one. Okay. So just give us a minute before we move on over there. Yeah.
Yeah, hi, my name is Maimona. I'm the event team manager for SNEV, and I would like to thank you for the wonderful talk and also for the interaction from the audience. We really learned a lot from your presentation. So thank you so much for accepting our invitation and also giving us all of this information. Yeah, it was really wonderful. And coming to our announcement, we have some exciting news. Maybe some of you already know, but SNEV is joining ICEV, the International Society for Extracellular Vesicles, as their early career researchers arm or community. So we would like to thank our incredible community for their trust and support. SNEV will continue along with ICEV to support and connect ACRs around the world with even more energy and impact. So stay tuned for more updates and celebrate with us this exciting news. Thank you.
Thank you so much, Maimona. And yeah, with that, I think we will conclude our talk. Thank you so much once again, Dr. Varas. We really loved your expertise here at SNEV, and we hope to cross paths with you again.
Thank you, thank you. And you are more than welcome to visit me here in Chile if you want to come.
Absolutely, yeah, sure. Thank you so much.
Yeah.
It's a little bit far.
Yeah.
Yeah.
Thank you so much.
Yeah,
All
I
right.
think we will have more talks with you as well.
Yes,
So yeah,
yes.
thank you so much for your generosity and for your time. Thanks.
Thanks.
Thank you.
Thank you for the invitation. Bye-bye.
Bye-bye.